So thanks everyone for having me today. I was asked to kind of give a high-level overview of what we as Thermo Fisher Scientific are doing around COVID testing, what we've done with some of the individual states that we've worked to scale and maximize testing, a little bit of what we have on the research side, and then I'll open it up to any questions people may have at the end. So with that, I'll kind of get started with the, typical disclaimer that this is for educational purposes only. If you do have specific questions, on testing or what you should do, we always recommend, reaching out to the CDC, your local state health departments, FDA, etc. So I'm a proud person to be representing Thermo Fisher Scientific today. I work for one of the divisions of Thermo Fisher Scientific, which is our genetic sciences division. We're kind of the applied biosystems brand of Thermo Fisher Scientific. And as an organization as a whole, we really have had a major impact in responding to public health crises throughout the years. So obviously we're deeply involved in COVID-19, but we've been involved extremely throughout the world with Ebola, H1N1, SARS, Zika, just to name a few. As an overall organization, we really have focused on the protection, where you hear about PPE and different things that we do to help our researchers and first responders. The analysis, some of the tools that we use. One of our Sanger sequencing instruments was actually the first one to actually sequence the coronavirus in Australia using one of our instrumentations. So it was the first one sequences outside of China. Detection, which is that rapidly develop, manufacture, and distribute the diagnostic tests for viral detection, which is what I'm going to dig into today. And then we are working with a lot of our customers that are trying to get vaccines for COVID-19. So we're working with them on the research side for what they need to support their vaccine candidates, as well as a lot of the clinical trial logistics. So overall, we do have an FDA-approved workflow from Thermo Fisher Scientific for COVID-19. It really is from a sample collection all the way through the analysis. There have been multiple reiterations of this as we've added on different sample types that you could work with, added on additional automation on the sample prep side, as well as increasing capacity on the real-time PCR side by being able to scale and really grow the number of tests labs can do per day. So what is our assay and what makes it so important? So actually the Thermo Fisher assay, we have 100% specificity to the SARS-CoV-2 gene. Genes have a tendency sometimes to mutate, especially viruses. So one of the reasons why our assay is so specific is we actually code it to 3 different regions of the COVID-2 assay, the targets, and we really focus on the regions that are more stable and don't have that mutation. So they have the lowest risk of mutation, which really helps us give that 100% specificity in our testing. I will jump through that, it's more boring stuff. And then really the end part of this, I shouldn't say it's boring stuff, it's super exciting stuff, but if you're not a science geek like I am, it may be a little bit boring. So I can answer questions at the end if you have them. But one of the main things that really pulls this whole workflow together is the interpretive software at the end that really helps you diagnose of, are they testing positive or are they testing negative? So like I said, as an organization, Thermo Fisher Scientific is a large organization with multiple divisions. We have about 75,000 employees, and it actually has been really phenomenal to watch our response to COVID-19. So we laughed that, all of this started the middle of February, which seems like it was so long ago of how long we've been dealing with this. But really to say all of these things have happened since the middle of February is absolutely amazing at how quick things have, how quickly companies, how organizations that are testing have really had to test their boundaries of how quickly can you scale. So right now, 50% of all the COVID-19 testing that's being done globally is using some product from Thermo Fisher Scientific. So it's either running our full EUA that I just showed you earlier, or it's using different equipment and pieces of the workflow that we have. So real-time instruments, could be master mixes and things like that. One of the things I like to really highlight is our scaling. So if we look at one of the components in a real-time PCR reaction, it's master mix. So master mix is one of the basic components you need for all real-time. Before COVID hit, to handle our global demand for master mix, we were manufacturing 500,000 reactions a week. So 500,000 reactions a week took care of all the research demand globally that we had for master mix. We are currently at 20 million reactions of master mix that we make a week to be able to handle COVID. So that's how quickly an organization that we've had to continue to build and scale. And we're continuing to go above 20 million, as this clearly is not going away anytime in the near future. And as more and more testing is coming on for back to school, back to work, we'll coin it back to life, there's obviously be more and more testing that's going to be needed. From a supply chain standpoint, anyone that's been involved in COVID-19 testing, they have heard, or I'm sure you've heard in the news of, we're running out of things in the supply chain. We don't have enough swabs. We don't have enough viral transport media. There's not enough kits out there to do the testing. One of the things that I'm very proud of is we really have ramped our entire supply chain to be able to fill those needs for our customers, and really being that trusted source that's able to work with our customers and make sure they can keep their testing going. And then, like I said, because we're vertically integrated, we're able to help really from soup to nuts as an entire workflow. And we have a full team of service and support field engineers and FAS, field application scientists, that are still going into the labs or working remotely via WebEx to help troubleshoot with our customers, train them, help with any needs, go in and make sure the instruments are up and running. So it really has been a phenomenal response of watching how everyone has come together to really scale this to meet the global demand. So when I talk about testing and the current approaches for COVID-19, it's kind of that scale. So you've heard in the news, or you've heard from different people, it's that how do you maximize testing? How do you get more and more samples in the labs? How do you get these things working? So when most researchers started out, they were really going with that manual step of running things in 96 well instruments, had a couple of full-time employees that were running it, and trying to get up to running 3,000 samples per 24 hours. Once you kind of got past that point, it became the tipping point of you're starting to throw more and more equipment, more and more people, more and more space needed to be able to expand the number of testing that you're doing. So the natural transition was to move over to the manual 384 well, where you'd increase your throughput by about double, but you can see the number of full-time employees. These are hands-on technicians that actually have to run the assay, accessioning the samples, moving them through the different workflows to be able to scale up to 10,000 a day. Some of our large labs, some of the big commercial labs that you're used to hearing, the LabCorp Quest, BioReferences, et cetera, of the world, they have a ton of third-party automation that they've incorporated in this and have tried to build, and kind of by brute force, being able to run over 12,000 samples a day. We have some labs that we're working with that are running 100,000 samples a day. So you can kind of imagine that amount of space and number of people they need to do that. The last one is really what we're moving towards, which is more of an integrated solution where we're adding and building a bunch of robotics in to have the ability to scale to, over 12,000 samples per day for this little workflow, but really cutting down on the number of full-time employees that you need to actually do it, because not only are the equipment and the reagents and the testing supplies, people are a rate limiting step in this COVID response of having qualified people trained to run molecular biology labs to be able to do this work. So one of the things that I'm really proud of from Thermo Fisher Scientific has been how we've actually responded to this. So in the early days, each state was trying to figure out what their plan was going to be and how were they going to be able to maximize testing, and how were they going to be able to make sure that they could take care of their state's needs. So one of the things we did as an organization is we proactively reached out to the government officials in each of the 50 states and saying, how can we help you? So as you saw earlier, we really have that end-to-end workflow for COVID-19. We have a ton of our equipment that is spread throughout lots of academic universities, biotechs, pharmaceuticals, clinical customers that are out there that, with shutdowns weren't being used as labs had to kind of close down for a bit before they were able to come back to work. So what we actually did is we worked with the states and gave them kind of an asset map of here's all the equipment that you have in the state, from the qPCR instruments that we talked about to the sample prep instruments. And it was amazing to watch as we reached out to some pharmaceutical accounts that were closed down, some biotechs, academic labs that were actually willing to loan their equipment to these testing labs so that they had that ability to scale. which was phenomenal to watch, just the overall team effort of everyone pulling together the resources that we had to maximize and really drive testing quickly. And as an organization, what we did was to make sure that we worked to redeploy that equipment, get it all trained, get the lab personnel scaled. So this really helped us, instead of trying to wait for inventory to come in, to get qualified people, to get them installed and training. We're able to enable testing in days at these accounts versus waiting for weeks, which is just awesome to see the teamwork of everyone coming together. So we do get a lot of questions on what should your testing strategy be, especially when you're looking at reopening, right? So a lot of the things that we will talk to our end users about is, a couple of these items here. Do you have a CLIA lab? Right, all of this clinical testing must be done in a CLIA lab. So we'll work to see if you have a CLIA lab and that's something that we can use. If you don't, should we recommend that you reach out to some other person that's doing, that's doing testing with other customers that can come in and they'll run those samples for them? Or is it worth looking to see if we can set up a CLIA lab in your facility so that you have that ability to do the testing? The second one is really, what is your testing strategy? Are you looking at just doing clinical samples? Are you starting to look at asymptomatic, from back to school, back to work? Is that additional testing you're trying to bring in? What we talked about earlier is really that sample collection process of, it's literally people that are taking swabs that are in a viral transport media that have to pull the swabs out to mark the tubes to get them to go through the section. So do you have the right sample collection process? Do you have the right people in place to be able to do it? Budget always plays a key role of, what are you looking at, kind of what types of things are going to make sense of bringing it in-house or outsourcing? And then what type of equipment do you currently have versus the need to buy more to help maximize what you're doing in the lab? So there's two main things that you see a lot coming up now. Media, people are looking at different ways. So you'll see different areas like asymptomatic versus symptomatic testing. So symptomatic testing are people that are actually showing displays of having COVID-19. They'll have the fever, they'll have the different symptoms that are showing it, where a doctor will say, you look symptomatic, we should really test you for COVID-19. There's a lot of conversations that what you're seeing is around asymptomatic testing. So you're starting to see this. Originally, we saw this in the retirement home or nursing home segment, where they were looking at that high-risk testing environment to do testing on asymptomatic to check. So right now, there's not really a lot of guidelines around asymptomatic versus symptomatic. So ourselves, as well as a lot of our labs, are working with the CDC and FDA on adding additional components of what asymptomatic testing would look like to ensure you can use the right things and the same things. Sample pooling was also something that came up, especially a couple months ago of, we have so many samples coming our way, how can we best maximize it? So the idea of pooling is, instead of testing one person at a time, you can actually pool together a number of individuals, test that group as a whole. If they test negative, then the assumption is that no one has it. But if somebody in that pool tests positive, you've got to go back and break that pool back down to figure out which person actually tested positive, to make sure you can alert the right authorities and that person, let them know that they have it and what we need to do to treat them. So sample pooling was a very hot topic because customers and labs were thinking that they would be able to pool 20 or more samples together. What we've been seeing is getting approval from the FDA is most researchers are looking at doing pools of 3 to 5 samples at a time. Quest is the first one that actually just got FDA approval for a pooling strategy, and they're at 4 samples per pool. So that's kind of one of the things you look at based on the number of positives you have in the environment you're working on. Is it worth just testing each one individually, or does a pooling strategy make sense to help you screen if you don't have as many positives that would make you have to go back and do it? So again, the FDA has sent out some guidance on it. Ourselves, as well as a lot of our end users, are working with the FDA to kind of see with our EUAs if it makes sense to add that, or if it's more of a laboratory developed test. So there's a lot of additional resources that you can use that are out there to help if you're looking to say, I want to set up testing, or what should I do and what's going to help me with my development. So I've put a couple of links in the deck, and I'm more than happy to send this out as a PDF to anyone that's interested after the call. But the CDC obviously has a lot of guidance on who should be testing, and there's links to the different state and local health providers here. The National Governors Association, because each state's kind of handling the responses differently of what, Pennsylvania would require, what New Jersey would require, are all kind of listed under that National Governors Association. And then there's a ton of information on the FDA website on what things have been approved for emergency use authorization. But then a lot of questions around, if I want to develop my own test, what do I need to do? If I don't have the positive controls to be able to submit it, how do I test it correctly? So a lot of, if you're looking to develop your own assay, things here for the frequently asked questions. Or if you're looking at a certain EUA and say, I want to follow it, but I want to make some modifications, that would make it a laboratory developed test. You'd have to work with the FDA directly on those. So outside of just COVID testing as an organization, we obviously do a lot on the research side to help our end users. So this is just kind of high level of what we have for viral research. So a lot of the products that Thermo Fisher Scientific has to offer are things in vaccine research with products, immune response research, looking at susceptibility, looking at viral typing, and then looking at epidemiology. That's a tongue twister for the end of the day, research in different areas. And as an organization, especially in the genetic analysis space where I am, we have a lot of equipment like next generation sequencing, microarray, Sanger sequencing and qPCR that customers are using every day to really do research on COVID-19 and getting an understanding of what this actually is, because it is a new novel virus that we're dealing with. And with that, I will leave it open if there's any questions.
So Kelly, I do have a question. You mentioned that one of the benefits that you guys bring is that you're vertically integrated, and so you have control over a lot of the supply chain side of it. And in the media, we hear a lot about sort of the congestion or the lack of tests at basically, I think your end users or your customer sites. And I wonder if you guys have have worked with any of your customers of how to better or more efficiently sort of process the testing on their end.
Yeah, no, we have. So it's actually been interesting to watch. So from all the different areas of the workflow, we've actually kind of been the opposite of salespeople, which cracks me up when you talk about it. But for some of the sample prep reagents, when we originally were working with it, we were running them in 400 microliter. So one of the first steps that we worked on one of our iterations as we've moved through with the EUA was actually being able to cut the amount of sample prep people needed in half. Which is funny because you're telling a customer like, hey, I want you to buy less of this so you can run double the amount of reactions. But doing that really helped them go from one kit that would run 1,000 samples at a time to have that ability for that same exact kit to run 2,000 samples at a time. So that's been one way that we've scaled. We've also worked with a lot of our end users on a weekly need. So instead of having a bunch of people placing a large bulk order that we're going to kind of sit in their lab and sit on a shelf, we've worked very closely with our customers to say, how much of this product, how many samples are you looking to run per week? And we've been able to ship those items out on a weekly basis. So this way, making sure that we're enabling the testing for everyone, that they have the amount of inventory that they need to run that week. They're not, for lack of a better word, we remember the toilet paper when you couldn't find toilet paper anywhere. We were very proactive in the very beginning to avoid the toilet paper thing where everyone bulk buyed items just to have it sit there where people that were in need actually needed it. There is a question in the chat. What's the primary bottleneck to testing throughput today? So Bruce, the answer I would give to you is really that upfront sample accessioning seems to be the biggest bottleneck. The equipment and running the sample prep and putting that stuff through the workflow seems to be fine. It's how do you handle the number of samples that you have coming in to be able to properly accession them, to be able to put them into the plates, to be able to run they're able to run them in the sample prep instrument. So we have some places that their sample accessioning, they only had 2 or 3 people that were doing it and could only really load, 396 well plates a day, which would really cut down the number of samples they could run. So what you do see is a lot of people throwing more physical bodies at that solution, where we have places that are running about 10,000 samples a week that are literally have about 13 people that are just doing that sample accessioning up front to be able to keep up with that throughput. So I would say the biggest bottleneck that I see right now is that upfront sample accessioning. So I don't know if these are all scientists on the call, if there's any non-scientists, but one of the things that I did put in the appendix, because we do get this question a lot, is the difference of the testing of what really is a viral testing and what does an antibody testing show. So this is a great tool that the White House put out that kind of talks to the difference of if you test positive for viral, which is qPCR, or if you test positive for antibody, or if you have a mixed result of both, what does that mean? So for any of the non-scientists out there, right, if you have a viral test that's positive, that means you have an active infection of COVID-19. What you will see is your virus will come up, and then as your virus load goes down, your body starts building antibodies. So you hear a lot now about antibody testing to see if you've actually had COVID-19. So if you test positive for antibodies, that primarily means you probably had COVID-19. The questions and a lot of the research that's being done right now, which is why I joke that even though we're doing, a lot of clinical testing with our assays, there still is a ton of research going on around COVID-19 is how long do those antibodies last? What type of response do you need to have to not contract it again? Does that wane with time, that maybe in 2 or 3 months, if you actually had another antibody test done, do you still show antibodies? So these are the things where you see people are looking at what their testing strategies are being. They're primarily focusing on right now that viral testing piece of do they have the virus, but you see a lot of places looking at that antibody piece to test of have they had it, but then it's going to be an ongoing of how often do you test for that, because how long did that antibody test happen for? And then you see a lot of confusion or questions that you get asked of, one great example is I test positive for the virus, but I test positive for the antibody. What does that mean? And it means you're still positive for the virus, but you're starting to build up your antibodies towards it. If you're, viral negative and antibody positive, that most likely means you had it, but now you've recovered and actually have antibodies towards it. Could I talk about error rates, false positives and false negatives? So a lot of the tests that are out there obviously have gone through a lot of rigorous involvement with the FDA, looking at data of how to avoid false positives and false negatives. What we're seeing is if you're not following all of the protocols to the T, it could potentially come up with false positives or false negatives. So we definitely encourage anyone that's running these assays to make sure that you're following the IFUs or instruction for use from any vendor that you're using it, and to make sure you have the proper training to be able to identify what those false positives and false negatives are. You also hear a little bit about this in the media with false positive, false negatives based on the way the sample was collected, right? Everyone knows at the beginning it was, we'll call it the brain probe, the nasal pharyngeal that had to go all the way up to your brain, that is very painful to able to get the swab. You now see people are doing nasal swabs. So is the nasal swab actually collecting the right amount? Did the person do it correctly? The joke we had was when you had people that were able to do that nasal pharyngeal swab on their own. There's no way anyone is jamming that as high up in your brain as your own person to be able to collect that sample correctly. So really, when you're looking at false positive and false negative conversations, how the sample is collected, how the sample is stored plays a role in that conversation, and then how the actual testing was run, and was it run the correct way, the way the protocols were written.
I have a question. Have you ever given this presentation or some version of it to children who might be interested in?
So it's actually kind of cool. So as an organization, right, we have a lot of our employees in multiple different divisions that have absolutely nothing to do with COVID-19. I feel like hence the reason why my hair is in a bun, because if you saw what my hair really looked like with my 3 inches of gray, no one wants to see that right now. So, a lot of the people that work for Thermo Fisher have been very in the weeds dealing with COVID-19, but we have a lot of people that haven't. And it's also, the children for like my kids are like, what do mom and dad do? So we actually just did our own little STEM thing as an internal thing for our organization, talking through, what are we as an organization doing for COVID-19? How have we really stepped up to help people? What does it mean? What types of things that we've done? And then we did the fun experiment at the end of pulling DNA extracts out of strawberries. So we've actually done that for some of our own people, but as an organization, we do a lot with different STEM programs. So if those are things people are interested in, and we have different groups within our organization that we're more than happy to kind of put things together for kids, because I will joke and say, I need a lot of scientists that want to be scientists when they grow up so they can come, work in the labs or work for our company to really help, continue doing what we all do and what we all love. However, my kids, I think, are into the arts. So I'm just, they see mom and dad as scientists and want to go in the opposite direction. I think that's the way it usually works.
Does anyone else have any questions for Kelly?
So my information's in the slide deck. If you guys do want to see a copy of it, let me know. I'll just email it to the group that put this together and they can share it. But any questions that you have or things that come up and you want to reach out or know who in the organization to talk to, reach out anytime and I'll actually get you in touch with the right people. And I appreciate the time. And, for those of you that are out there in the forefront of dealing with COVID-19 testing or, all the different, frontline workers, and I will include anyone that's feeding me food right now as part of my frontline worker. Thank you so much. But I appreciate everything you're doing. And, the take home is stay safe. We'll get through this and we're going to get through this together.
Thank you so much, Kelly. And thank you, Thermo Fisher Scientific, for sponsoring the session today. And thank you everyone for attending. Excuse me. And we have some other programs coming up later this evening, including I'm gonna start up roundtable starting at 5:30, and then a session about designing a COVID vaccine from home at 6:30. But thanks again, everyone.
I had a question, St. Louis.
Sure.
Do you have problems getting supplies for your machines and reagents?
We've been really good at keeping up with the supplies. Like I said earlier, we've been working on weekly allocations, and I sit on calls all day long of allocating product to our customers. So it's really working with them of what they actually need to make sure that we can ship it. So far so good. We've been really great at supplying, but we've also done a great job of continuing to scale as demand increases.
In the chat, I put the archbridge.us in there. That's something we started in St. Louis that we have FDA initial import credentials, and we can bring that in officially if you guys need help on any of those reagents or gloves and things like that.
Okay, great. Thanks.
If you look at it. Yeah, thank you.
Great.
All right, well, with that, I will jump to my next COVID-19 call with an end user. So thank you so much, and I appreciate the time.
Thank you.